Morning, everyone. Welcome to session 5. We have an exciting lineup of speakers for you. Um, it's a pleasure to introduce one of my oldest friends in the oak business, Jeannine Cavender-Bares. Jeannine is a professor of organismic and evolutionary biology at Harvard University and the director of the Harvard Herbarium. Jeannine's work focuses on the physiology and evolutionary dimensions of plant ecology that influence community assembly and ecosystem function. She's particularly interested in the genetic and evolutionary basis of variation in oak phenotypes and spectral properties that can advance remote sensing of biodiversity, particularly detecting change through time in consequences for ecosystems. Let's give a big round of applause to one of the leaders in our field, Jeannine.
Hi, everyone. Thank you so much for being here and coming 8 in the morning. I'm going to talk today about some work that's been going on for a while, trying to understand the ecological diversification of oaks and using spectroscopy to test adaptive evolution across environmental niches. Let's see if I can get this. It's not advancing. Try this. There we go. So I will start with this slide that I've shown many, many times, which shows that using forest inventory data in the United States and Mexico, oaks are the most abundant and the most diverse woody group in both the United States and Mexico. And so they are ecologically important and evolutionarily important. And when I was a graduate student back in the 90s, and I started trying to ask questions about how plants relate to their environment, it became clear to me that I really should be focusing on something important. I wanted to have ecological relevance. And I looked around the Harvard forest where I was where I was in graduate school doing graduate work, and red oak made up 60% of the forest. So that's a very abundant species in that particular region of the world in New England. And so I started working on red oak and measuring its response to drought and learning all kinds of things about oaks by studying red oak at the Harvard Forest. But I knew that the oaks were very diverse. Walter Judd had come to Harvard and told us about all the oaks that were in the southeastern United States. And I knew that they were very diverse in Mexico. And I went down to Mexico to see if I could work with Miguel Martinez Ramos on oaks in Mexico. And I ended up working in the southeastern United States, in, in mostly in Florida, and just became obsessed with the oaks because they were so diverse and doing such different things ecologically. And so this whole time I have really wanted to understand the answer to this question: why are oaks so abundant and diverse? And I think we spent years with Paul and Andrew and other folks in Susanna and Antonio trying to answer this question. And that really led to the phylogenomic reconstruction of the oaks that Andrew led us through, and recognizing that we had a pattern of sympatric parallel adaptive radiation with the red oaks, the section lobati, and the white oak, section quercus, radiating adaptively and in parallel across the continent during the Eocene Oligocene transition when the earth was cooling and the tropical taxa were drying out. And there was all this ecological opportunity to diversify, spread across the continent like people explained yesterday, and move down into Mexico, where at that point the mountain chains were rising and it created new opportunities for diversification, and rates of diversification sped up, which is why there's so many oaks and so much diversity in Mexico. And this has been an absolutely fascinating story to live through and be part of. It's just been one of the joys of my life, uncovering being part of uncovering that history and that story. And the oaks, can you get this to advance? So the oaks, because they are so important, so diverse, so abundant, and because they went through this whole process of diversification in North America, it became very clear how important the diversification process is for understanding community assembly. So I come largely from an ecological background where the big question is, how do species assemble into communities? There are these species sitting there in some species pool, and they assemble into communities based on ecological rules like environmental filtering and competitive exclusion. But it became very clear that you can't actually understand community assembly without understanding the deep evolutionary history. And it also becomes clear that if we need to understand the rules of community assembly, if we're going to put our planet, our living planet, back together, restore and regrow our planet in the Anthropocene, where we are really changing literally everything. And so the oaks have been a phenomenal model system to really understand the ecology and the evolutionary history that is critical for understanding how species assemble into communities, how they generate the ecosystem functions we all depend on, and understanding that deep history and the nuances of the ecology and the physiology really provides insight to how for us to manage our planet, to build a habitable planet in the face of so much change. So I remain dedicated to the oaks, and in large part because of all of you. I've had people over the course of my career make fun of me for focusing on the oaks because it's, oh, what is that? Just a genus of plants. And without this society with 20 countries and people from all over the world really focusing on this, sometimes it can be hard to justify why you spend so much time studying something that seems, that to other people might not seem as important as it is. So thank you, all of you, for everything you do to make this community and to show the world how important this group is. Okay, so now on to some fun. So over the last number of years, I've been going around collecting leaves and the stems to look at wood anatomy and leaf properties. I'm not going to talk about the wood anatomy today, things like freezing tolerance and the way vessels and tracheids look in oaks. But I'm going to focus today on the leaves. And this is at the Shields Oak Grove, UC Davis. I saw Emily here. And so we collected in the wild and in Arboreta in California. We went to England and collected in Sir Harold Hillier Garden. And thank you, Alan, for dedicating so much of your life to making this such a wonderful collection with reliable identities. And then Beatrice and Gerard hosted me at Pouillet. It's such a wonderful collection. It's amazing work that you have done over all this time. And there's Beatrice in the tractor helping me collect branches from all of these species. We got wood samples and leaf samples from so many species. And then, of course, my friends in Mexico, who have many, many times driven me all across the country collecting species here. And Susana Valencia, I think, has identified every single species I ever collected here. And this is a nice story that Beatrice and I wrote about this most recent trip that we did in 2022. And I'm sorry that I missed the pre-conference tour where we retraced some of this. And there's Ricardo and Oscar and Antonio, and we had such a wonderful trip collecting across Mexico. And of course, also went to Puebla and took advantage of this amazing garden that Marcella and Alan have spent years cultivating. And so over this period, I've collected many leaves and stems and pressed them for herbarium specimens, but also took spectral measurements, spectral reflectance measurements using a spectroradiometer on both fresh leaf tissue and dried leaf tissue. And the reason for doing this, and Mariana will explain more in her talk subsequently, but spectral reflectance is on the y-axis of this graph in the middle, and I don't know if I have a pointer, but there we go. The y-axis of this graph is reflectance, and it goes between 0 and 100. This is the visible range. So this is what your eye can see. So your eye sees that there's not very much reflectance in the blue or the red because that's where plants are absorbing. There's a bit more in the green, which is why plants look green. And then there's the near infrared, where plants don't absorb very much. And then there's the shortwave infrared. These dips are where water is absorbing, so there's not much reflectance there. But then there's all this incredible variation in the shortwave infrared that your eyes cannot see. But it picks up information about the anatomy and the structure and the chemistry of the plants. The shortwave infrared particularly gets its structure, gets its scattering of light. So it gets at all kinds of anatomical features of plants that your eye can't see. And it has allowed us to both predict the chemistry and functional traits of plants, but also to differentiate them across the tree of life. And so some years ago, I led a working group to the National Institute of Mathematical Biology and Synthesis. And at that time, this is Dudu Mireles, who did his PhD with Paul Manos. And he led the analyses on this. But all these people brought together their data, their spectral reflectance data from plants across the world. And we had to harmonize the data because of all kinds of issues of people using different instruments and different optical setups. Dudu did all that. And we found that there was a high degree of phylogenetic signal, meaning close relatives have more similar spectra than you would expect at chance. But different wavelengths showed different amounts of signal for different lineages. And we can also use spectra to predict a whole suite of plant traits. And this has largely been done with fresh leaves. So we use machine learning models like partial least squared regression. But we found it also works well for dried leaves. In fact, sometimes it even works better. And so for fresh leaves, you can predict this whole set of traits, pigments and sugars and carotenoids and carbon fractions and micronutrients, nitrogen and phosphorus. For dried leaf spectra, you can predict a subset of that, but sometimes even better. So you can actually use herbarium specimens and measure functional traits from herbarium specimens by measuring the reflectance. And so, Paul, this was before you talked to me last night. I had this picture in here already. Here's Paul and Kathleen Pryor in the Duke Herbarium, and this is my new role as a director of the Harvard University Herbarium. They have a massive oak collection, and it hasn't yet been digitized, so we're working on that. So we've been measuring the spectra of herbarium specimens, and so many people have gotten excited about this. We recently had, we started this international herbarium spectral digitization working group, and people from 3 continents met at the Harvard University Herbarium in May. And we developed protocols that are publicly available that we've agreed upon so that we can aggregate the data at the end. Okay, so the goals for what I'm going to show you now, and I guess I'll have to go quickly, is to investigate the use of spectra from fresh and dried leaves to understand evolutionary processes, to quantify rates of evolution of traits, and to determine how traits evolved in relation to environmental niches. So we used herbarium records and pulled environmental data from those locations for all the major lineages of the oaks. And we could do that because of the arboreta that you folks have curated over your course of your lives, because I didn't actually go to Asia for this. And use the Hippodoll 2020 phylogeny that Kieran Althouse updated for us. And this initial analysis shows the phylogenetic signal. If it's a red blob, it means there's phylogenetic signal on the reflectance at that wavelength. One thing you see is that across fresh leaf spectra, there's phylogenetic signal within the oaks all across the spectrum. The shortwave infrared goes away in dried plant tissue, and I don't know why, except that water really is important for the structure because the turgor pressure in the cells is critical to the structure that causes the scattering in the shortwave infrared. So that might be related to why we don't see phylogenetic signal in the shortwave infrared. And it's interesting, I'll keep going. On the left are fresh, and on the right are dried leaves. We fit evolutionary models, so a Brownian motion model, an Ornstein-Uhlenbeck adaptive model, an early burst model. And for every wavelength, for both fresh and dried spectra, the Ornstein-Uhlenbeck model, the adaptive model, fits the best. So that's what we use. And here you can just see the spectra on top of there. So it's only where the water is absorbing that something else might be going on. Then we develop models. This is the validation data. So when you develop these machine learning models, you use in our case, we use 75% of the data to train, calibrate the model. We use 25% to validate it. This is just the validation data. So a lot of data goes into building these models for all of these traits. These are statistical models, machine learning models to predict traits. They don't always work. And so we just use the models. We just use the trait predictions for which we have accurate results. A lot of traits are correlated. For fresh leaves, we can measure many more traits because we can use physical models to also predict pigments and various attributes of the plants. For dried leaves, we have fewer traits that we chose to predict, but this shows the correlation among them. Okay, so here's some major results, and I'm just going to show you for now leaf mass per area and thickness. And we find, this is across all the lineages of oaks around the world. So this is about 200 species, I think. And it differs a little bit for fresh and dry. But leaf mass per area and leaf thickness increase with higher elevation and with coarser or poorer, better drained soils and with greater evaporative demand. So when the going gets tough, the leaves get tougher. They get, more mass per area and thicker leaves. Okay, so now I'm going to show you cellulose. Oops. So what's interesting here is not actually the lines, which there's a little bit of relationship, but what's starting to blow my mind about cellulose is that the white oaks are largely under the mean, and the section quercus is under the mean, whereas section lobate is above the mean. And in all these cases, the white oaks have low cellulose. And I had never, so I've done a lot of work measuring plant function in American oaks, and I always find these patterns of convergent evolution where they show similar physiology, similar hydraulic function, similar photosynthetic rates, similar leaf properties. And here we're finding actually the white oaks are doing something really different. And so what is cellulose? It's this building block. These chains of cellulose molecules form microfibrils that form macrofibrils that reinforce the cell walls. They're really critical for plant growth because without the cellulose in the cell walls, when the turgor pressure builds up, that allows the cell to expand. So it's important for cell expansion and growth. It's also related to leaf toughness. So it's related to herbivory. It's related to leaf lifespan. So have you ever noticed that white oaks just don't have as long leaf lifespans as the red oaks do? I had never put this together, but it's hard to find white oaks with long-lived leaves. It affects litter decomposition. Microbes don't like to chew on cellulose. It's really yucky. So all these things that I hadn't thought about before are related to cellulose. This is a study from the Neotropics Barrow, Colorado Island from Kero Kitajima, showing that leaf cellulose is the key determinant of leaf fracture toughness, or how tough leaves are, which is related to other ecological attributes. Okay, so let me just show you some. This is a Bayesian model that fits an Ornstein-Uhlenbeck There are multiple optima for traits, and it just shows every time there's a major regime shift in the trait for leaf mass per area and leaf thickness. To orient you, this is section Quercus, so these are the white oaks, these are the red oaks, and there's Ceres and Cyclobalanopsis in here, Ilex. Okay, so there's been some major regime shifts for leaf mass per area and leaf thickness. The phylogenetic half-life, meaning the amount of time it takes to get halfway to a new optimum, is about 1.73 million years for leaf mass per area. It's 0.26 million years for leaf thickness. Lignin shows something similar to that, and here I've kind of annotated the different lineages. But look at cellulose. Like, it's just different in the white oaks. And look at the phylogenetic half-life. It's 20 million years. So what's going on? The white oaks just are fundamentally different in their leaf construction. If we take the first principal component of all the leaf traits, these are the white oaks here. They're just different from everything else. This just shows a principal components analysis with the major lineages. So you can see Ceres, Cyclobalanopsis, Ilex, Lobate, Ponticea, Protobalanus, Quercus, Virentes. The major lineages, the red oaks and the white oaks, encompass huge trait space, leaf trait space. And this helps explain why they occur in so many different niches. And there's a lot of overlap in the trait space that they occupy. This is for fresh leaves. You see the same thing in dried leaves. You see that the major lineages occupy a large amount of trait space. The smaller lineages occupy narrower phenotypic space. And another thing we see from this is looking at rate. So this is the sigma squared. This is the rate parameter in the Ornstein-Uhlenbeck model. And interesting, you see rapid rates of evolution in leaf function and leaf phenotypes in the red oaks, in the Mexican and U.S. southwestern oaks, which matches what Andrew Hipp and our team found earlier, that the rates of evolution speed up in Mexico because of the rise of the mountain chains that led to rapid diversification. So we picked that up in these leaf phenotypes as well. So in summary, I'm still working on this. Hopefully I'll actually sit down and write this paper this spring. Where we are right now is that we find that leaf mass per area and leaf thickness evolve in relation to critical environmental niche axes with higher leaf mass per area and thicker leaves in harsher environments. And this is across all lineages of the oaks. The major clades differ in leaf chemical composition, especially cellulose, which is linked to cell expansion and growth, to herbivory, to leaf lifespan, to litter decomposition. It might explain some interesting things like, the white oaks are more resistant to pathogens, to fungal pathogens. The red oaks are more invasive. You notice that? Like, they grow faster. They're pushy. They take over when there's an opportunity. But they're really not very resistant to pathogens. Look at what's happening with oak wilt in North America. Oak wilt is killing all the red oaks, but not the white oaks. The white oaks have staying power. We do know that their wood anatomy is different. And the way that they form tyloses, their immune system's different. So they have staying power that may be the expense of their growth. And that maybe over time has led to their coexistence, where you have the pushy group and then you have the resistant group. And they coexist because pathogens come in and knock out the ones that are not resistant. Okay, we're also finding that different leaf traits evolve at very different rates, and that major clades are radiated to span large realms of trait space. So that's what I have to tell you today, and I'm glad for your questions.
I was just wondering, does it matter where on the oak tree you collect the leaf from, like the sun leaves versus shade leaves? Does that make any difference at all?
Sure, it absolutely makes a difference. And if you were to just measure leaves within a single tree, you'd find higher leaf mass per area in the sun leaves than the shade leaves. We tried to collect in sunlit branches. That's one way to try to standardize. And then we also tried to get many individuals per species, so we're capturing variation.
Um, Thank you for a fascinating talk. I'm over here. I had a question about leaf shape. The typical leaf shape that we associate with oaks, with those beautiful lobes, you see that they're very widespread across even different sections. But yet in Mexico, we don't have that leaf shape as seen as often. And I was wondering if there's something about that shape that provides an advantage in colder weather or different radiation, solar radiation, and on light, or
You know, that is a question that has fascinated many people, including folks in my lab. And this study is not getting at leaf shape. One thing that's interesting is that so there's a couple things. The perimeter to leaf area ratio is influenced by how much lobing there is. And it's also highly related to hydraulic conductance. So the higher the perimeter to leaf area ratio you have, the higher the hydraulic conductance, the faster the growth. That's an empirical relationship, but it may have to do with the way the veins are able to spread out over the leaf and move water quickly and supply gas exchange and maximize photosynthesis, because moving water is all about gaining carbon. So it may promote faster growth. Also, when you have a smaller leaf with less lobe, it's going to lose less water. And so you might get thicker leaves that concentrate photosynthesis with thicker cell layers, but are less susceptible to water loss. And so I'm still trying to make sense of the talks yesterday on Pertussis cygnus, because it's different when you look within species than across species. And also there's this phylogenetic baggage where, the plants might start with a history where they were in one environment. They may have evolved into a new environment, and they sort of start with a leaf shape that their most recent ancestors had, and then they evolve from there. And so you can get, within the oaks, you can get all of these uncoupling of relationships that you might normally expect across the tree of life, the plant tree of life. So that I'm rambling now, stop.
Thank you very much. I do enjoy this talk. You talk about this relationship between leaf LMA and harshness. What types of harshness? You had one with elevation, but what do you mean by harsh?
Okay, so more evaporative demand and more and drier conditions. That's one. Coarser soil and elevation. So elevation's tricky, right? Because it can be warmer at the bottom and cooler at the top. But you also have less moisture at the top. At least you have less soil moisture. And then there's going to be issues with clouds and so forth. Like cloud forests are going to be different. So I'm going to say drier, more well-drained, more nutrient poor. But you also notice that's a general trend. And if you were to take any point in any of those axes, there's a huge amount of variation. So it's the same with the leaf economic spectrum, where you draw these nice axes and then you think you can explain everything with that. But if you look at any one point in that spectrum, there's a huge amount of variation, and you really have to dig in and see what is going on in each place. we did a study in Arizona up and down the mountains there and actually found that leaf phenology was critical to adaptation along the elevation gradient. And it was because at the bottom of the mountain where it was hottest, they became drought deciduous. The oaks became drought deciduous. At the top of the mountain, it got really cold, so they became winter deciduous. And in the middle of the mountain, they were evergreen because that was the most benign condition in the middle of the mountain. So phenology is really a key trait that oaks use to deal with different kinds of stress, and they can time it based on when the unfavorable season is occurring. So that's just a nuance in one part of the world. This was trying to cover all the oaks and try to come up with general patterns. So that's the best I can say for now.
Yeah. Great, thank you.
Brief catch up with Jeannine at the coffee hour. We have an announcement from Ryan.
Morning. Just a couple quick announcements. First one is for ISA certified arborists. If you are looking to get CEUs for the conference, I'm going to have a sign up sheet in the silent auction room. ISA is doing things a little bit differently now. It used to be you sign up for each talk. They're doing kind of a bulk thing now. So I'm going to have one sign up sheet for all certified arborists. You fill in all your information and then I'll turn that in when we get back. Second thing is about the silent auction. Silent auction is going to start at the first break this morning. I want to thank those of you that donated items and those of you that didn't. Got to dig into your wallets. Okay. So bid often, bid high. We'd like to see a little competition there. There are some instructions at the bottom of the sheets. There's some instructions on the top as well. Please bid in US dollar amounts. That doesn't mean you have to pay that way. For those of you that be paying in pesos, we can do that at the end of the auction. For those of you that win, we'll accept that. And the auction is going to close at 6:30 sharp on Wednesday. So 6:30, get your bids in before then. Last thing is those of you that donated some items, I still need a few of you to fill in your information at the top with your name and what the item is, so that we can correctly get that listed. So that's it. Thank you.
You know, we forgot to thank Jeannine for her talk. Was awesome. Okay, our next presentation is virtual, so I'm hoping the technology works out here, but here's the introduction for Dr. Mariana S. Hernández Leal. She's a postdoctoral researcher at Harvard University, working with Jeannine in the ASCEND group. She specializes in genetic molecular evolution, physiology, and spectral trait biology, using experiments, models, and observations to study biodiversity across scales. Her work focuses on how trees adapt to challenges like drought, extreme temperatures, and salinity, blending statistical programming in R with GIS tools to tackle these questions. She has also explored how spectra can link phenotypic and genomic data, shedding light on natural selection and adaptation. Let's welcome Mariana, and I don't know how to control it from here, but you got it? Thank you.
Hi everyone. My name is Mariana Hernández Leal. I am a researcher associate at Harvard University, working in the lab with Dr. Jeannine Cavender-Bares, and today I will be presenting the results of our most recent publication, which appeared this September in the New Phytologist issue. The paper is titled Foliar Spectral Signatures Reveal Adaptive Divergence in Section Virentes Across Species and Environmental Niche. And basically, I'm going to talk about how to use spectral data as a high throughput sequencing technique and how to compare it with genetic data in order to reveal adaptation in different species. So, as an introduction, I'm going to start to talk about how in evolution variation can come from two main processes that we know as isolation by distance, which is related to the neutral forces of gene flow and drift, and isolation by environment, which is driven by natural selection and adaptation. So we all know that neutral forces usually shape genetic variation, while selection often shapes phenotypes. But in reality, these forces act together all the time, so it is hard to separate which one is responsible of the diversity that we see today. In recent years, with the advance in high throughput sequencing, we have seen an exponential increase in our ability to quantify genetic variation across individuals, populations, and species, which has given us the great opportunity to identify, like, for example, SNPs associated with certain traits, but also like to do GWAS analysis, the construction of high density maps, haplotype maps, or even phylogenetic identification of candidate genes. But measuring phenotypic variation has been always been a challenge. For example, to assess functional traits in leaves, like some traits that are associated with physiological features like gas exchange, fluorescence, or others associated with morphology like thickness, mass, or pigments, you need different instruments and a lot of time to measure each trait in every sample. So, leaf reflectance has been proposed as a high throughput way to measure plant phenotypes. So we all know that leaves reflect and absorb light in ways that have been shaped by evolution. So by analyzing their light patterns, we can learn about important functions without measuring each trait individually. High resolution spectral measurements of leaves and canopies allow us to indirectly and non-destructively assess key structural and chemical traits which are closely linked to physiological and biochemical properties of plants, and also associate them with specific genotypes. So here we all love oaks, and we know that oaks are an excellent model for studying how neutral and adaptive evolutionary forces interact and shape genetic and phenotypic diversity. Because they are a key component of temperate ecosystems, they have a wide geographic distribution across diverse environments and for complex interactions with other species. They are really important for the ecosystems. They also interact with closely and distance related species within the same genus, and each is adapted to different microhabitats, allowing multiple species to coexist within the same range. Moreover, oaks hybridize quite easily, and yet despite sharing similar environmental conditions, these species remain distinct, even if they have like a big loss of gene flow between them. So we start working with live oaks, that is a group of 7 closely related monophyletic species distributed across temperate to tropical environments in North America. These species show patterns of speciation in sympatry, like the ones that are in Florida, allopatry. For example, we have Quercus brandegae that is only found in the Baja California Peninsula, and parapatry, and display unique genetic and phenotypic structures shaped by their evolutionary history. By using these traits derived from leaf spectra, we can characterize the phenotypic structure of this lineage in a similar way to how we analyze genetic variation and then compare difference between genotype and phenotype. So, to study how these neutral and adaptive forces have shaped the evolution in this lineage, we use an integrated framework consisted of: first, we use dry leaf spectral data from samples that have been really well preserved by Dr. Jeannine Cavender-Bares, some even when she was doing her PhD. So this, by using these dry leaves, we were able to predict functional traits that then were compared with genetic neutral data to assess like a genetic distance versus a trait distance approach. And then we used environmental variables to test isolation by distance versus isolation by environment in order to determine if the environment was driving the adaptation in those traits. Finally, we integrate a phylogenetic through PGLS models to help evaluate adaptive divergence across species. So in our results, first using only the spectral data, we did a confusion matrix using a PLSDA analysis using only the spectral data for these 7 Quercus species in the subsection Virentes. So for example, here in this matrix, we can see that the columns represent the observed identities of each species, while rows indicate the predicted identities only using the spectral data. Values along the diagonal represent the percentage of individuals that were accurately classified within each group, while values above or below diagonal represent the percentage of misclassification. So for example, we can see that Quercus minima and Quercus guilleminata have some percentage of misclassification between them. So here we can see the typical representation of genetic structure, and we have also at the right the phenotypic structure. We use these 7 species to create these graphs, for example, using 11 nuclear microsatellite data for the genetic structure, and for the phenotypic structure, we use 6 spectral predicted traits from the leaves using the same individuals to compare the genetic and phenotypic grouping between these species. And we know like different patterns between these two genetic and phenotypic structure. For example, in northwestern Mexico, where Caicus fusiformis and Geckos oleoides populations are parapatric, here represented with these red lines, the genetic data shows that they share alleles between these 2 species, so there is gene flow between them. But if we compare that with the phenotypic structure obtained by the derived traits, we see that there is a clear differentiation between these populations. So we propose that isolation by environment have shaped this phenotypic difference between these 2 species, even if they have a lot of gene flow between them. And for example, Quercus fusiformis exhibits the highest freezing tolerance among the Virentes species, while Quercus oleoides is highly vulnerable to freezing. So, despite the gene flow between these populations, isolation by environment may have prevented populations of Quercus oleoides from expanding further northward due to strong isolation by low temperatures. Another example is, like here we can see in blue, is the absence of clear phenotypic structure in these species of the Caribbean that are consisted of Quercus geminata, Quercus minima, and Quercus virginiana that lives in Florida Peninsula, Quercus sagrana that is only found in Cuba, and Quercus oleoides and the parts of the Caribbean. So we know that Quercus sagrana and Quercus oleoides are sister species. So there is like a hypothesis that Quercus sagrana have been originated from these populations of Quercus oleoides in Central America. But then they have like subsequent integration from Quercus geminata and Quercus virginiana. So despite geographically restricted gene flow, particularly in our current era of high sea levels, this may be attributed to retained ancestral polymorphisms between these different species. So they have, maybe they have a similar phenotypes because they share some environmental variables between them. So using the same traits derived from the spectral data, we can obtain phenotypic distance and compare that with genetic distance, product of genetic sequencing, to quantify which traits are different among different lineages and how adaptation have affected them. So these matrix, for example, represent PST plotted and compared versus FST or genetic distance. PST is quite similar to QST, but is to compare with wild populations in which we cannot make like real QST comparisons because we don't have like common garden experiments. So for example, using leaf traits derived from spectra, the sympatric species like Quercus geminata, Quercus virginiana, and Quercus minima, that you remember they all are distributed in the Florida Peninsula, showed significant differences in their PST pairwise distance for almost all traits. For example, Quercus virginiana and Quercus geminata are different in all those traits except for cellulose, while Quercus minima versus Quercus virginiana had significant difference in their PST pairwise distance for leaf mass area, thickness, cellulose, and hemicellulose. And linked for the pairwise comparison between Quercus caminata and Quercus minima, that are these two populations that, if you remember the structure graph, they were clustered in the same genetic group. Only cellulose showed significant higher values of PST than FST. So meaning that cellulose is a really important trait for distinguish environmentally these two species. And because the wavelengths reflect how certain compounds or functional traits use light, we can use those same wavelengths as a high true-up traits to identify areas of the spectrum that have been selected in different lineages. So for example, this figure represents the wavelengths that had the highest VIP values or variable of important values that we obtain after doing the PLSD classification model using only the spectral. The orange vertical lines represent wavelengths that were used as traits to calculate this PST pairwise distance, and these metrics are divided into the visible, the near infrared, and the shortwave infrared spectral regions. So we now can compare and see how evolution and adaptation had affected these different regions of the spectra that are associated, for example, with chlorophyll in the visible region and structural traits in the short and the near infrared, and see how these different species had evolved between that just simply by adaptation. But our main objective is not only to determine whatever genotypic and phenotypic differences exist within these species, but also to identify the ultimate causes of such differentiation. So, is it product of the environment through adaptation, or is just isolation by distance? It is important to know that both the neutral and adaptive forces can drive genetic and phenotypic differentiation. So, for example, these three closely phylogenetic species are sympatric and they coexist in the same landscapes. Moreover, they share some gene flow between them. Since environmental factors are highly correlated with geographic distance, it is necessary to disentangle the contribution of each variable to phenotypes while controlling for the effects of the others. So we use a peer the model to observe that in the three sympatric species, phenotypic differences are better explained by environmental conditions than by geographic distance among individuals. Furthermore, we identify which regions of the spectrum are most influenced by environmental variables. For instance, in this species, the soil moisture index is a key factor in determining their microclimatic distribution that is strongly influenced by the access to the water table, as illustrated in the corresponding diagram. So we can expect that the environment is really important for adaptation of many of these traits. For example, low levels of lignin are related with cold tolerance. So species distributed in northern latitudes should show a lower content of lignin than southern ones. However, it is crucial to consider that related species are not statistically independent to their shared evolutionary history. This means that closely related species tend to share more traits due to their common ancestors, which generate covariance among them that must be accounted for in the analysis. Using a PGLS framework allows us to incorporate this phylogenetic covariance into the models, which provide more robust and unbiased estimates of how traits are associated with environmental gradients. In other words, PGLS helps disentangle whatever observed patterns are the result of adaptation to the environment or simply due to shared ancestry among species. So, for example, Quercus brandegae, that is related with Quercus falciformis, actually is the sister species of Quercus falciformis, doesn't share this low lignin because adaptation had through the desert had molded this trait between these two populations. So, for conclusions, by integrating high true-up sequences with the spectral derived phenotypic traits, we can bridge the gap between genotype and phenotype, offering new insight into how environmental and geographical factors shape evolutionary trajectories. And this approach not only advances our understanding of oak evolution, but also highlights the broader applicability of spectral data in studying adaptation and natural selection across plant lineage. So I want to thank you for coming to my presentation, and also I want to thank you to my co-authors and to Ascend, that is this wonderful group that have been studying spectral data for a long. So thank you for coming, and if there are questions, I'm open.
We have plenty of time for questions. It's Mariana. I think she is.
Yeah, I'm here.
Mariana, we can't drum up a question. Can you show Mariana? Oh, Mariana, let's see you. Yeah, there she is.
Sorry about my look.
You look great. Okay, so well, let's thank Mariana. Okay, we'll move on to our third speaker. We have the pleasure of having Akiko Satake is with us. She's a distinguished professor at Kyushu University in Japan. She uses mathematical and computational approaches to study diverse aspects of biological systems. She's interested in evolutionary dynamics of mass seeding and biogenic volatile organic compound emissions in Fagaceae tree species. Let's welcome Akiko.
I'm Akiko Satake from Kyushu University. It's a great honor to be introduced by Professor Manos. Thank you very much. It is first time to visit Mexico, and also it is first time for me to join this conference, IOS. I joined a pre-conference tour, and after joining that tour, I'm so impressed by amazing diversity of oak species in Mexico, and also amazing enthusiast of acorn. And I really enjoyed the atmosphere of this conference with a strong connection between society and academia. I sincerely appreciate the lot of effort organizing committee to organize such a wonderful conference. So today I will talk about the evolution of biogenic volatile compound emission in Fagaceae. I took this picture when I visited Yunnan province in China, where there is another hotspot of biodiversity of Fagaceae species. So from this forest ecosystem, a huge amount of volatile organic compounds called BVOC emitted. So yesterday, Roderick introduced Japanese haiku. So I want to introduce one beautiful haiku by a famous poet, Santoka Taneda. Wakeitemo, wakeitemo, aoyama in Japanese. The meaning in English is go deeper and deeper, yet still the blue mountain. So when the forest emit a lot of BVOC, it looks blue because of sunlight scattering. So I'm pretty sure that he saw the mountain where there are many oak species. And BVOC includes diverse compounds, and isoprene accounts for about a half of BVOC emission. And isoprene is emitted highly in hot summer. And it is reported that emission of isoprene is effective to induce heat tolerance due to the protection of photosynthesis system by emission of isoprene. And the second most largest amount of BVOC is monoterpene. And monoterpene, known as a communication molecule that signal danger to neighboring plants, and damage by herbivora increase emission of monoterpene by dozens of times. So these BVOC emission affect climate because part of BVOC becomes aerosol and contribute to the cloud formation, which thereby inducing cooling effect due to sunlight scattering. So that result in the change of solar radiation, temperature, precipitation, so which feedback to the plant survival and reproduction. So there is a bidirectional feedback between the plant and climate. So to understand such a bidirectional feedback, as a first step, we study about the evolution of BVOC emission in Fagaceae. Forest ecosystem is not composed of a single species, but rather it is assembly of species exhibiting different BVOC emission activity. Some species are high BVOC emitter, but there are species that do not emit BVOC. And studying Fagaceae species is very interesting because this Fagaceae has a great variation in BVOC emission, especially isoprene emission. So this plot is the isoprene emission rate for many different plant family and Fagaceae here. And you can see great variation here in Fagaceae species. And Fagaceae plants also contribute to BVOC emission at global scale because of its wide distribution in the world and also a large biomass. And which species emit isoprene? Yes, section Celcas species emit a large amount of isoprene. So this is a photo of the Japanese white oak, Celcas serrata. This species is known as high emitter of isoprene. But the species in Cyclobalanopsis, like Celcas glauca, almost no emission. And other genus, the Rhizocarpus edulis, in the genus Rhizocarpus, low or no emission. So there is a great diversity of isoprene emission in Fagaceae family. So I'm very interested in how has the diversity of isoprene emission evolved. So there are two possibilities. The isoprene emission have been gained during the course of evolution, or the ancestor used to emit the isoprene, but during the evolution, that phenotype lost. So to test which is more likely, we performed a comparative analysis of terpene synthase genes called TPS. So this is a terpenoid biosynthesis pathway. And hemiterpene, that is isoprene, and monoterpene, G-terpene, sesquiterpene, all are terpenoid. So to synthesize these terpenoid, the plants need terpene synthase called TPS. And TPS contribute to the synthesis of all of these compounds. And in general, the TPS form a large gene family, but we don't know how many TPS genes are present in Fagaceae species. So we compared TPS gene across 8 Fagaceae species. The 5 species from genus Celcas, and 1 species from genus Rhizocarpus, and 1 from Castanopsis, and the last one is from genus Fagas. There are several high-quality genome in Fagaceae, but the number of species in which high-quality genome available is still limited. So only 8 species, but in future, maybe number of species we can target will increase. So first, we analyze the synteny relationship of the genome of 8 species. And all of species we study have 12 chromosomes. And the genome structure, the arrangement of gene, are highly conserved between Celcas species, Rhizocarpus, and Castanopsis species, but Fagas crenata is exception. So very different synteny. So the gene arrangement may have occurred in this genus in the past. So somewhere in this genome, there are TPS gene. So we extracted TPS gene from this genome using the conserved domain known for TPS. And also we used the intron exon arrangements that are conserved for TPS gene. And finally, we selected a gene that is long enough, that are long enough. And as a result of extraction of TPS gene, we obtained in total the TPS gene from 20 to 60 TPS gene for each species. And Castanopsis and Fagaceae species, relatively no number of TPSG, but Quercus glauca and Quercus rubra, a lot of TPSG. And TPS gene family are divided into subfamily from TPS A to G. And this is a gene phylogeny. And we can see that TPS A and B subfamily diversified. So some sequence, some TPS should be isoplane synthase that are associated with isoplane emission, but we don't know which gene are isoplane synthase. But previous studies show that isoplane synthase have evolved in the TPS gene subfamily. So we look at this gene family more in detail. And here we included the tomato and Arabidopsis TPS gene. And in tomato, the previous study identified isoplane synthase. And this star indicate ISBS in tomato. So this clade could be the target clade where the Fagaceae ISBS included. So we named this clade as TPSB target 1. And we also found the twin clades that are near to the target 1 clade. And this clade could be the candidate for isoplane synthase. So we named this clade target 2 and target 3, and then we calculated isoplane score. So isoplane score here is a number of conserved amino acids that are specific to isoplane synthase. So this is the 3D structure of the isoplane synthase in populus albus. And there are 4 important amino acid sites that are important for isoplane emission synthase. And especially the 2 phenylalanine that are facing each other are important because these 2 sites limit the size of the substrate that entering to the reaction center of this enzyme. So we calculated isoplane score for the target sequence of Fagaceae. And we found that all of gene included in the target 1 clade, the score 1, very low score. But in the clade, the target 2 and target 3, we found the gene with the highest score, the score 4. The 2 genes of Quercus robur have score 4 in the target 2 clade, and 1 gene in Quercus robur, the score 4 in target 2. And in target 3, we also found the 3 gene in Quercus robur, robata, and rubra, the maximum score of isoprene. And this high score, isoprene score, is consistent with the high emission of isoprene of these 3 species. So this means that isoprene score is a good index for the actual isoprene emission. So we are interested in the evolutionary trajectory of isoprene emission in Fagaceae. So to understand whether ancestor emit isoprene or the no emitter, we predicted the amino acid sequence of ancestor using codon model. And we found that, so this is a result. The N1 is the most ancestor. So N1 amino acid sequence, the isoprene score is only 2. And N2 and N3, the score is also 2. But at the node of N4, isoprene score increased from 2 to 3. And finally, at the node N6 and N4, N8, it became score 4. And interestingly, we found that so this acquisition of the phenylalanine, the last important amino acid, this acquisition occurred independently twice in these plants, N6 and N8. And we also used plant site model to test whether the acquisition of this phenylalanine amino acid is under the positive selection or not. But and we found the evidence of positive selection. So the evolution of the acquisition of phenylalanine site is under the could be adaptive. And we also monitor the seasonal expression pattern of TPS gene in the target 2 and target 3 clade. And we found that the gene in target 2 clade, very low expression. So the gene in this clade is not functional. But the gene in the target 3 clade show clear seasonality. It is highly expressed in summer. So we think so this gene could be the target, the candidate of ISBS in Fagaceae. So that's the final step. We predicted the enzymatic, we performed enzymatic characterization of ancestral protein. One of the good things to study isoprene is that we can associate the gene sequence with the phenotype, isoprene emission. So we artificially synthesized ancestral TPS sequence and subjected to enzymatic characterization using E. coli expression system. So we used the node 7 and node 8 sequence and use E. coli expression system, and then tested, examined whether that these two ancestral proteins showed isoprene emission phenotype or not. And we found that the sequence at node 7 did not show any monoterpene emission, but the sequence at node 8 show isoprene emission. And instead, the sequence in the node 7 show monoterpene emission, but node 8 did not show monoterpene emission. So this means that the ISBS has evolved from the monoterpene synthase, and it becomes isoprene synthase. Okay. So this is I summarize the evolution of isoprene emission in Fagaceae. So this is a species phylogeny, and the duplication of target 2 and target 3 clade occurred very long time ago, before the divergence of Fagaceae. But at that time, the isoprene score was low. But about 56 million years ago, the phenylalanine site, important site, gained, and isoprene score became 4. And gain of the phenylalanine site occurred again in the limited species of the section Celcas. So the evolution, so isoprene synthase evolved from monotypic in subgenus Celcas approximately 56 million years ago. And this kind of evolution would change the quantity and composition of BVOC emission. So now isoprene accounts for a half of BVOC emission. So, but maybe in the past, the monoterpene might be the majority. But maybe in future, due to the evolution, the BVOC composition in the air can change. So to understand what would happen in future, I'm very interested in studying more species in Mexico and North America, where species diversity is huge. So during this conference, if you are interested, would be very happy to discuss with you about this topic. So now we are running a project about this topic, the feedback between climate and plant. So we have a homepage. So if you are interested, please visit. And at last, I'd like to thank all of collaborators. I always enjoy working with a wonderful collaborator. Thank you very much for everyone. And thank you very much for your attention. That's all I want. Oh.
Yeah, it's great. I really enjoyed your talk. Thank you. I kind of wasn't clear about one thing and then a follow-up. One is, what do you think is the advantage to the plant, to the tree, to have these emissions? I could see its impact, but I'm not sure what the selective advantage. And sort of related to that is, do you see any geographic trends or ecosystem trends in the species that are showing high emissions, and therefore it's cooling things off? Any kind of patterns that give you a little bit of insight about the selective factors on this trait?
So adaptive significance of isoprene emission is still in argue, but there is a report showing that it induces heat tolerance. So when they express ISPS in Arabidopsis, normally wild type can survive lower temperature, like 40 centigrade. But if isoprene is emitted, it can survive in 50 centigrade. So definitely isoprene emission induce the heat tolerance. But so I think oak tree also emission of isoprene would be have a adaptive role to protect photosystems under heat and harsh condition. But still, the evolution of isoprene emission is mysterious because the species that emit isoprene and the species that do not emit isoprene coexist in the same environment. So more study needed. Yeah.
I was wondering, well, thank you for a really great talk. I love hearing about this system. I was wondering if you've looked at gene expression pattern with plant damage for some TPS genes to show that isoprene has summer emission, but do some other ones have induced emission by herbivory? Or induced expression?
I think so. So during our monitoring, we did not record the infestation level, so we cannot answer that question. But I think previous studies show that these genes activated when they are infested. Yeah.
Thank you.
Do organic volatile compounds contribute the greenhouse effect?
Greenhouse effect. So in the introduction, I briefly explained about the cooling effect. So BVOC have a role in to induce cooling effect by sunlight scattering. But on the other hand, it has the warming effect because during the so complicated chemical reaction, after emission of BVOC, that can increase the methane that has a great warming effect. So there is a contradicting effect, cooling or warming. So yeah, still in ague. So we need to explore about that effect. So, but in a polluted region, the warming effect is stronger. But natural vegetation area, the cooling effect is stronger. So the balance between the two is very important.
I have one for you. Did you notice any casual correlations between evergreen species and deciduous species, or temperate versus tropical species
Causal correlation with
with emissions?
emission.
Yes.
The Dani et al., the previous study showed that deciduous tree is more likely to emit isoprene, and and fast growing tree also more likely to emit isoprene. But there are still variation. So it's not clear cut between the two. Mm.
Wonderful. Okay, time for the last talk of this session. We have the speaker. Okay, let me introduce you. The last speaker is Antonio Reyna Perez. Antonio studied chemistry at the Faculty of Chemistry at UNAM, where he became interested in instrumental analysis and chemical analyses, and how this field can provide information about natural processes. He pursued his master's degree in earth sciences with a focus on environmental sciences, where he developed a project that sought to determine the relationship between alkane synthesis in oaks and soil water availability. His research brings together ecophysiology, analytical chemistry, and biogeochemistry. Now, in his doctoral program at UNAM, he aims to determine the ecological fate of these compounds associated with oaks from two different sections of the genus. Let's welcome Antonio.
Thank you. Thank you. Estoy muy emocionado de estar aquí. I will proceed in Spanish if you want to use the translation. Estoy muy emocionado de estar aquí y compartir con ustedes un poco de lo que hemos hecho en mi proyecto de doctorado. Quiero comenzar hablando de los biomarcadores, que es un término que se utiliza para denominar una gran variedad de compuestos orgánicos que se utilizan para inferir condiciones del pasado terrestre, ya sea para determinar procesos u organismos. Como se ve en esta figura, pues la mayoría una característica de estos compuestos es que son estructuralmente complejos, ya que una condición para servir como biomarcador es que se mantengan inalterados en el registro geológico por miles de años. Como el título de mi presentación indica, nosotros nos centramos en los alcanos lineales. Los alcanos lineales son compuestos formados exclusivamente por carbono e hidrógeno, cuya longitud está dada por el número de carbonos. Estos compuestos se encuentran dentro de la cutícula de las hojas, esta barrera que protege a los tejidos foliares contra el estrés ambiental, tanto biótico como abiótico, y estos compuestos dentro de la cutícula nos pueden ofrecer información valiosa a diferentes niveles. En primera instancia, al estar dentro de la cutícula, estos compuestos nos pueden indicar sobre las adaptaciones que hacen las plantas hacia las condiciones ambientales. Específicamente, los alcanos, al ser compuestos hidrofóbicos, se sugiere que tienen mayor importancia en evitar la desecación de los tejidos. Además, como mencioné, también se utilizan como biomarcadores en estudios de reconstrucción paleoecológica, bajo la generalización de que las plantas leñosas generan una mayor proporción de los alcanos de 27 y 29 átomos de carbono, mientras que las herbáceas generan una mayor proporción de los alcanos de 31 y 33 y que este patrón se mantiene inalterado en el registro geológico durante miles de años. Sin embargo, nosotros consideramos que esta generalización deja fuera ciertos procesos ecológicos que pueden incidir en la composición de estos compuestos. En primera instancia, como todo compuesto sintetizado en las hojas, ¿no? Su síntesis está condicionada por la respuesta a las condiciones ambientales y la ontogenia, lo que puede generar variaciones en la síntesis de estos compuestos a lo largo de la vida de las hojas. Una vez que las hojas han cumplido su ciclo de vida, sufren un proceso de senescencia que conlleva la reabsorción de los nutrientes que sean móviles dentro de los tejidos, lo cual puede afectar indirectamente a la composición de los alcanos y se genera así un segundo compartimento, la hojarasca. Una vez que la hojarasca llega a la superficie del suelo, se genera un nuevo compartimento llamado mantillo, en el que se encuentra una variedad heterogénea de residuos vegetales en diferentes grados de descomposición. Una vez que los compuestos presentes en los residuos vegetales, que ya no se puede identificar la identidad de los compuestos asociados a ciertos residuos vegetales, se dice que estos se han incorporado a la materia orgánica del suelo, donde también pueden estar sujetos a la descomposición o al aporte por microorganismos y raíces. Si recuerdan, al principio mencioné que una característica de los biomarcadores es que deben de ser estables a lo largo del tiempo. En el caso de los alcanos, pues son compuestos relativamente recalcitrantes porque son hidrofóbicos, no tienen grupos funcionales que estén activos. Sin embargo, hay una gran variedad de microorganismos, principalmente bacterias, que han desarrollado la maquinaria metabólica para poder utilizar estos compuestos como fuente de energía o carbono. Entonces, esos procesos se pueden dar tanto en mantillo como en suelo. Pese a la importancia de todos estos procesos, son pocos los estudios donde se analicen toda esta ruta vertical. Eso es algo relevante. Con estos 4 compartimentos se genera lo que nosotros llamamos ruta vertical de cambio químico o vertical pathway o chemical change, donde se acoplan procesos que pueden modificar la estructura o composición no solamente de los alcanos, sino de todos los compuestos generados en las hojas. Pese a la importancia de todos estos procesos, son pocos los estudios donde se analice de forma sincrónica los 4 compartimentos. Generalmente se analizan alcanos en hojas verdes y en suelo. En este mapa se muestran los estudios donde se analizan hojas verdes contra otro compartimento, generalmente suelo, y como pueden ver, pues no hay representación ni del territorio ni de las especies mexicanas. Entonces, aquí viene la sorpresa, ¿no? Nosotros decidimos enfocarnos en estudiar encinos. México es un país encino diverso y los 2 principales linajes de encinos en nuestro país son la sección Quercus y la sección Lobate, encinos blancos y encinos rojos, que como nos actualizó la doctora Susana Valencia ayer, incorporan 86 especies por secciones. Además, se sugiere que estas 2 secciones tienen tolerancias diferentes a la sequía. Pero como nos han mencionado compañeras el día de ayer, estas 2 secciones presentan distribuciones simpátricas en el territorio mexicano debido a la historia evolutiva de estas especies. Entonces, todas estas características nos abren la posibilidad a estudiar especies filogenéticamente cercanas con diferentes adaptaciones ambientales bajo las mismas condiciones y el efecto que tendrían sobre la síntesis y conservación o transformación de los alcanos lineales. Por lo tanto, el objetivo de este proyecto fue analizar la composición de alcanos a lo largo de la ruta vertical con el fin de responder 2 grandes preguntas. Primero, si hay convergencia en la síntesis dentro de secciones, y la segunda es si el patrón de síntesis de alcanos en hojas verdes se mantiene inalterado hasta llegar a la materia orgánica del suelo. La hipótesis general inicial que planteamos es que si los alcanos inciden en la tolerancia a la sequía, y este es un rasgo conservado entre las secciones, esperaríamos que las especies de encinos blancos presentaran una mayor concentración y un favorecimiento hacia la producción de alcanos más largos, puesto que son más hidrófobos, y que el proceso de descomposición incidiera de forma más intensa en residuos de estas especies debido al condicionamiento de la comunidad microbiana. El sitio donde trabajamos se encuentra en la cuenca de Cuitzeo, en el estado de Michoacán, donde tenemos un clima templado con precipitaciones marcadas de junio a noviembre y donde los bosques de encinos son la vegetación predominante. Particularmente nosotros trabajamos en el cerro llamado El Remolino, que se encuentra al suroeste de la ciudad de Morelia. En un fragmento forestal de este cerro encontramos la coexistencia de encinos rojos y encinos blancos. Tuvimos Quercus castanea, rojo, y Quercus desertícola, Quercus calófila y Quercus laeta. De cada especie seleccionamos 6 individuos y colectamos muestras de los 4 compartimentos en puntos que consideramos importantes para tener una instantánea de la dinámica de los alcanos a lo largo de la ruta vertical. Una vez colectada cada una de las muestras, lo que hicimos fue la extracción de los alcanos asistida por ultrasonido y la identificación y cuantificación de los alcanos la realizamos mediante cromatografía de gases, acoplada a espectrometría de masas. Voy a presentar los resultados comenzando por los encinos rojos. La estructura de las siguientes 4 diapositivas es la misma. Del lado izquierdo para ustedes, está las gráficas de concentración de cada uno de los alcanos identificados. En código de colores están cada uno de los compartimentos, y del lado derecho está un diagrama de Venn que muestra las similitudes de alcanos presentes en cada compartimento. Lo que quiero resaltar, empezando con Quercus calófila, es que, contrario a lo que uno esperaría, la mayor concentración de alcanos la determinamos en hojarasca, las barras rojas, lo cual nos sugiere que el proceso de senescencia en esta especie no es tan intenso o que está favorecida la producción de alcanos hacia el final de la vida de las hojas de esta especie. También determinamos la presencia de 3 cadenas adicionales a las observadas en hojas verdes, en este caso de menor número de carbono, y el alcano de 29 átomos de carbono presenta una mayor concentración que la de 27, aunque no significativamente mayor. En Quercus castanea determinamos el mayor rango de alcanos de desde 18 hasta 33 átomos de carbono. En esta especie sí determinamos una mayor concentración en hojas verdes como se esperaría, y determinamos la adición de 2 cadenas en los compartimentos posteriores a hojas verdes, en este caso de mayor número de carbono, y también vemos que la cadena de 29 átomos de carbono es mayor a la de 27. Ahora los encinos blancos. En Quercus desertícola es donde vimos la menor variedad de alcanos. En este caso también observamos una mayor concentración en hojarasca. Entonces, procesos similares a los que se observan en calófila deben de estar sucediendo durante la formación de este compartimento. Y en esta especie determinamos la adición de 6 cadenas diferentes que no se observan en hojas verdes, en este caso de menor número y mayor número de carbono. Y curiosamente, la concentración del alcano de 29 átomos de carbono y de 27 son más cercanas. En el caso de Quercus laeta, en este caso también observamos la mayor concentración en hojas verdes y la adición de 3 cadenas también de mayor y menor número de carbonos a las observadas en hojas verdes, e igual mayor similitud entre las cadenas de 27 y 29 átomos de carbono. Entonces, los procesos particulares de cada compartimento están incidiendo en la composición de estos compuestos. Para profundizar en ello, realizamos un análisis de componentes principales para cada especie, utilizando los alcanos presentes en todos los compartimentos. Están señaladas los compartimentos de las muestras de suelo, y un análisis de ANOVA sobre cada uno de los componentes arrojó diferencias significativas entre las muestras de suelo y las muestras de hojas verdes, ya sea en uno o de todos los componentes principales. Entonces, podemos responder una de las preguntas iniciales: si el patrón se mantiene inalterado hasta llegar al suelo, y pues vemos que no, los procesos están incidiendo. Para comparar entre especies, ya que como vimos no hay consistencia en los rangos, tomamos el rango entre 23 y 31 átomos de carbono, que sí es consistente entre las 4 especies. Aquí les presento la suma de la concentración de los alcanos en este rango para las 4 especies en los 4 compartimentos. En primera instancia vemos que en hojas verdes y en hojarasca no hay consistencia en la concentración a nivel seccional, ni siquiera entre las 4 especies. Entonces, esto no es indicativo de que hay divergencia en la síntesis de estos compuestos y en el proceso y en cómo afecta el proceso de senescencia. Después, en hojarasca, lo que observamos es que hay convergencia en la concentración. Hay la misma concentración en las 4 especies. Sin embargo, por lo que vimos en los compartimentos previos, no podemos indicar que esto sea consecuencia de la identidad de las especies, por lo que creemos que debe de ser consecuencia de la heterogeneidad del mantillo, debido a que el material se encuentra en diferentes grados de descomposición, lo cual en este caso está haciendo una convergencia en concentración. Y por último, en suelo volvimos a encontrar diferencias. Sin embargo, viendo los compartimentos intermedios, no podemos decir que podemos convenir que no es directamente trazable a lo que está pasando en las hojas, que es lo que comúnmente se hace. Si nosotros solamente observáramos los extremos, pues podríamos considerar que las diferencias observadas a nivel de hojas son las mismas que se están observando en suelo, lo cual nos llevaría a hacer una inferencia errónea, lo que resalta la importancia de los compartimentos intermedios. Quiero que nos centremos un poco en las hojas verdes con el fin de responder la otra pregunta inicial: si hay convergencia en la síntesis de estos compuestos a nivel seccional. Y no, vemos de hecho que hay mayor similitud entre encino rojo, encino blanco, encino rojo, encino blanco. Entonces, retomando la hipótesis inicial, pues en este caso no observamos una mayor producción de estos compuestos de forma consistente en los encinos blancos. Puede que ahora se pregunten, entonces, ¿estos marcadores son aplicables en encinos? Y yo respondo que probablemente sí, pero no como el perfil completo, sino con un cociente. Si recuerdan, señalé la relación entre 29, el alcano de 29 y 27 átomos de carbono. Entonces construimos un cociente con estas concentraciones y determinamos a nivel de hojas que sí había una diferencia seccional. Los encinos rojos presentan valores más altos de este cociente que los encinos blancos. Posteriormente, al avanzar en nuestro gradiente, vimos la disminución en el cociente de los encinos rojos, pero no vimos cambios en los encinos blancos, hasta llegar a suelo, donde convergieron las 4 especies y ya no son discernibles entre sí. Entonces, estos cambios observados en los encinos rojos nos hacen pensar en la hipótesis de la amplitud funcional, que indica que la descomposición está mediada por qué tantas funciones pueden hacer la comunidad microbiana. Y aparentemente la comunidad microbiana asociada a los residuos vegetales de los encinos rojos tiene una mayor maquinaria para hacer uso de este tipo de compuestos que los encinos blancos que permanecieron inalterados en los 4 compartimentos. Entonces, ahora surge la duda de esto es trazable o se puede aplicar a más encinos y bueno, perdón, antes, ¿no? Retomando la hipótesis inicial, pues tampoco observamos que los encinos blancos presentaran un mayor efecto por la descomposición. De hecho, fueron los encinos rojos los que lo presentaron, al menos con este cociente. Ahora, ¿qué tan aplicable es hacia otros, extrapolarlo a más especies de estas dos secciones? Desafortunadamente, son pocos los estudios donde se analicen alcanos en especies de encino o que se indique específicamente qué especie es, pero con 5 especies adicionales que encontramos, pues vemos que hay consistencia al observar este cociente entre el alcano de 29 átomos de carbono y de 27, con los encinos rojos presentando valores más altos que los encinos blancos. Entonces, quizás este cociente podría ser alguna clase de indicador quimiotaxonómico, no lo sabemos, falta analizar más. Entonces, un poco de recopilación, vimos que hay los procesos particulares de cada compartimento tienen efecto, ¿no? Modifican la composición de estos compuestos incluso antes de que lleguen al suelo. Entonces, concluimos, sí hay variación a lo largo del gradiente vertical. Vimos la incorporación de nuevas cadenas, tanto más largas como cortas. El cociente C29, C27 podría ser un rasgo conservado a nivel seccional, pero ¿cuál es la función ecológica de estas relaciones? Es algo que hay que averiguar. Y en general, pues es necesario profundizar en los procesos de cada compartimento para utilizar estos biomarcadores de forma precisa. Y hay mucha gente a la que quiero agradecer, todas las instituciones que me han apoyado, los profesores, el doctor Antonio González, el doctor Fernando Pineda de la ENES Morelia, CONACYT por la beca a lo largo del posgrado, y quiero también agradecer a la sociedad por organizar este evento y por el grant que me otorgaron, por el cual puedo estar aquí presente y platicar con ustedes. Muchas gracias.
Thank you. Do you know, can you use soil and alkanes to differentiate oaks from other species, even if the profile doesn't always work for among species of oaks?
No hemos, el objetivo final del análisis de este tipo de compuestos justo es observar en suelo, obtenerlo de suelo y hacer inferencias. Sin embargo, con el perfil completo no encontramos una forma de discernir. Cuando calculamos el cociente pensamos que podría servirnos, pero como vemos, en suelo se pierde la señal que sí alcanzamos a ver en hojas. Entonces, no quiero decir que no, porque creo que podríamos todavía buscar otro tipo de relaciones. Hay otro tipo de indicadores que se utilizan que toman en consideración la concentración de mayor número de alcanos entre otro tipo de alcanos. Quizás explorando otros índices podríamos llegar a eso, pero con lo que llevamos al momento, no.
Pero otros árboles que no son cuercos.
No hemos explorado más allá de este género por ahora.
Gracias.
Thank you.
Okay, we'll be back at 10:20, but there's an announcement from Tonio.
Thank you, Paul. Yeah, a few announcements. Please check your emails. You should have received this morning important information regarding the gala dinner tonight and also the field trips tomorrow. So make sure that you understand everything, all the instructions. And if you have any questions, please let us know. The gala dinner is going to be at the Centro Gastronómico, which is not in the hotel, it's in the downtown area. It's about a 20-minute walk from here, or you can take a taxi from here. You can order a taxi at the hotel reception, and it takes 5 minutes to get there by taxi. For the comeback, it's the same. There will be taxis available outside or near the door, so you can take a taxi and come back here. For the field trips tomorrow, please be sure that you know which trip you're in and follow the instructions. There are some itineraries for each field trip. If you have any questions, please let us know. Also, the silent auction is open, so you're invited to go and see the items. Some items have like a minimum amount to start bidding, so take that into consideration. And well, I think there are very beautiful and interesting things there that you can buy. As Paul said, the coffee break is going to be shorter because we are late. So please come back at 10:20 for Antoine's talk.
That's good.